And many of you may have been accepted to medical school and you're waiting to arrive and start your first week of class. Some of you are pre-med students. Some of you are med students watching this. And so I made this video because this is what I wish I would have done before I started medical school. I'm in my first week right now, and the biochemistry is ridiculous. And in the biochemistry, we're basically looking at an overview of glycolysis, glycogenesis, glycogenolysis, and gluconeogenesis, and how all of those things affect metabolism. And a lot of the enzymes in those 4 different metabolic pathways are being referred to over and over and over again, and they perform, and they're acted on by different things and perform different functions. And it really would have been helpful, I think, if I would have just memorized every enzyme and every substrate in those pathways. And so what I did actually for some of my classmates and myself is I made a little study guide. So actually I borrowed this picture. Let me pull up so you can see the credits on it. I borrowed this picture from the Harper's Illustrated Biochemistry. So Harper's Illustrated Biochemistry 29th edition. And I actually took and blacked out all of the enzymes in the pathway, and I did that so that I could fill in the blanks, and then I can use the original as my answer key to see if I've got it right or wrong. And you can download that if you're on my blog. It should be just right down below as a link to go ahead and download. But what I wanted to do is for anyone that's looking at it and wondering what do all of these lines and squiggles and minus signs and plus signs and what does all this stuff mean, I just want to go ahead and go through it really quickly so that you can understand it. So when glucose goes into a cell, it can either go into a liver. In a liver cell, it can be done a lot more things with. The liver kind of is the control hub for metabolizing glucose. Or it can go, like, say, in a muscle cell. In a muscle cell, a hexokinase or some hexokinase would act on it. In a liver cell, glucokinase would act on it, and they would both perform the same function just at a different speed. And their function is they're going to turn it into glucose 6-phosphate. And so the liver cell using glucokinase would turn it into glucose 6-phosphate a little bit slower than, say, a muscle cell or a brain cell. Once it's in glucose 6-phosphate, it gets transformed into fructose 6-phosphate. Now it's important to note that anytime we see these curved outward arrows going from one substrate to another, it represents an irreversible enzyme reaction. So glucokinase and hexokinase will only go in this direction. They won't go backwards like other enzymes. So once you get glucose 6-phosphate, the enzyme that catalyzes this reaction, which I think is an isomerase, but I'm not entirely for sure, it goes both ways. Depending if the concentration of glucose 6-phosphate is higher, it drives it this way. If the concentration of fructose is higher, it drives it that way. So this is a reversible step. This one is irreversible. Then fructose 6-phosphate, through an irreversible reaction with phosphofructokinase, comes and converts into fructose 1,6-bisphosphate, which goes through a reversible reaction with glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this gets converted. So fructose 1,6-bisphosphate is 6 carbons, and it gets split apart into 2 3-carbon molecules. Now dihydroxyacetone can actually easily be converted straight into glyceraldehyde 3-phosphate by an enzyme called triose phosphate isomerase. I'm just going to put triose phosphate isomerase, ASE. Or it can be acted on by glycerol 3-phosphate dehydrogenase, which this is a reversible reaction to produce glycerol 3-phosphate. Moving on from there, the G3P, glyceraldehyde 3-phosphate, gets converted reversibly to 1,3-bisphosphoglycerate, then to 3-phosphoglycerate, then to 2-phosphoglycerate, then finally to phosphenolpyruvate. And then the phosphenylpyruvate is converted into pyruvate. So when we have pyruvate, well, first that's done by pyruvate kinase, and pyruvate kinase is another irreversible step in this downward ladder. So now we've had 3 irreversible steps from glucose to pyruvate. Now pyruvate, if the cell doesn't have enough energy, it's going to go into the mitochondria and convert into acetyl-CoA and go through the Krebs cycle, the tricarboxylic acid cycle. But however, if there's plenty of energy in the cell and it doesn't need to convert into acetyl-CoA, it can actually be acted on by pyruvate carboxylase, and what will happen is CO2 will be added to it and it will turn it into oxaloacetate. So remember in the Krebs cycle there's already an oxaloacetate, and so you start with one, you go through, and you end with one. Whenever you go this route, you may already have oxaloacetate and you're using pyruvate to create another one. So now you've doubled your amount of oxaloacetate, and it doesn't need that much to run the Krebs cycle, so it actually will convert oxaloacetate into malate, and the malate aspartate shuttle will ship it out of the cell, and then it will be converted back into oxaloacetate. And then through phosphenylpyruvate carboxykinase, it can be converted into phosphenylpyruvate. So we've just went through this irreversible step, and we went through a new metabolic pathway to reverse it. It's important to know that for the most part, this doesn't happen in cells other than the liver, but it does happen. So we have converted it to phosphenylpyruvate. Once the concentration of PEP, or phosphenylpyruvate, has increased tremendously, it will drive the reaction all the way back up to fructose 1,6-bisphosphate, and right here. So once you have this, you can actually, because phosphofructokinase is only one direction, you can actually use fructose 1,6-bisphosphatase to remove one of the 2 phosphate groups on it to convert it to fructose 6-phosphate, which can be reversibly go back to glucose 6-phosphate. And then glucose 6-phosphatase can reverse the irreversible glucokinase reaction and recreate glucose. So this is important because whenever your body's running low on energy, you can actually break down amino acids and use them to create new sugars. So actually, for example, alanine, cysteine, glycine, serine, threonine, and tryptophan can all be converted into oxaloacetate, which can then go through this pathway and then be converted into a sugar. Asparagine and aspartate can also, alanine, cysteine, glycine, serine, threonine, and tryptophan can all be converted to pyruvate. Asparagine and aspartate can be converted into oxaloacetate. Aspartate, phenylalanine, and tyrosine can be converted into fumarate, which can be converted into malate and then into oxaloacetate. And then isoleucine, methionine, threonine, valine can be converted into succinylcholine and go, of course, into this pathway. And so you have a lot of different amino acids that can be broken down and converted into sugars, and this is done by the liver whenever your body is running low on when your blood sugar is low, essentially. So how is all this regulated? So that's what all the squiggly lines are. The squiggly line means whenever this is high in concentration, what does it do to these? So this actually slows down glucokinase and hexokinase whenever glucose 6-phosphate is high. It just acts allosterically on those enzymes. Fructose 6-phosphate increases, whenever this is at a high concentration, it increases phosphofructokinase, whereas a high concentration of ATP will stop the phosphofructokinase. So if the cell has plenty of energy and it doesn't need to make any more energy, it doesn't want to go down through the glycolysis pathway, so it's an inhibitor, which would come over here when AMP is high. So whenever the ATP has been used up, AMP will cause the drive this way, whereas the lack of AMP would drive it this way. And so anytime you see a minus sign, these are things that whenever they're in high concentration, they will inhibit the enzyme from acting. And so that's what all of the squiggly lines are about, the inhibition. It's the control of this, so the body maintaining a balance of exactly what it needs and exactly when it needs. And so you can see all of these squiggly lines and exactly how they inhibit or enhance the action of the enzymes. And so what I did was, like I said, I made a little blank chart, and you can download this from my blog, and the link should be in the comments. But you can use this to fill in the enzymes and practice them. And then, of course, I would also just recommend memorizing this whole thing before you start medical school. I promise you, you'll thank me for it. Now, the one thing I'd encourage you to do is while you're going through and memorizing all of this, as you get each section, let's say you've memorized all of this, as you get each section memorized, go through and see if you can logically figure out why each thing would either inhibit or uninhibit a specific enzyme. Like, why would a high concentration of fructose 6-phosphate allosterically activate the phosphofructokinase? And so if you can logically go through that and say, well, and then ask yourself, what do I think would be a stronger inhibitor or activator? So in this case, ATP, I'm thinking, and don't take this for gospel, but I'm thinking ATP would be a stronger inhibitor than fructose 6-phosphate would be as an activator. Now, with that said, and even though this comes from a biochemistry textbook, I'm not entirely sure that all of these are correct. The one that actually confuses me a little bit is, why would a high concentration of fructose 1,6-bisphosphate deactivate the fructose 1,6-bisphosphatase? Because you would expect, oh, high concentration, okay, now it's going back in this direction. So the concentration, I'm assuming, would have to be high for this reaction to move this way, but I could be wrong. I could be completely off base on this. So it might be, the Km the Michaelis constant for fructose 1,6-bisphosphatase may be such that it acts at very low concentrations on this, and so once you get a high concentration, it turns it off. I could be wrong though, and it would make sense in that case, because a high concentration of bisphosphate would drive the reaction downward if this enzyme was off.